Okay, so leveraging plant microbe synergy for growing healthier crops. I like to start off my presentations with a bit of a quote just to kind of set the stage, set the context of the session that we or the content that we're going to go over today. The preservation of fertility is the first duty of all that live by the land. There is only one rule of good husbandry: leave the land far better than you found it. George Henderson, The Farming Ladder, 1944. Highly recommended book, by the way. So, first of all, I want to thank you for having me here. I just want to quickly introduce myself. Um, as Julia mentioned, my name is Jo Tobias, and I own and operate Root Shoot Soils, which is a company that specializes in regenerative soil and living compost. I have been since 2015 collaborating with farmers across British Columbia, Alberta, Saskatchewan, Manitoba, and Ontario. So my work centers on helping land stewards like yourselves develop a deeper relationship with your soil. In my efforts to regenerate degraded landscapes, I discovered that well managed compost, keyword well managed, can serve as a powerful tool in kickstarting ecological processes that drive nutrient cycling. It drives decomposition, which are essential in regenerative agriculture since you're relying less on synthetic inputs. So, really focusing on microbial community to do that work for you. Before immersing myself in soil health, my background was in computing science. And software development, where the core question in that environment was always how can we do things better. So I analyzed data, I optimized company operations using knowledge driven software and software that I helped write to basically help diverse industries make sense of their information. And that mindset of continuous improvement followed me when in 2010 I transitioned from computing science to eventually finding permaculture along the way and eventually soil ecology. So a pivotal moment came during my visit to my family in the Philippines, where I saw firsthand the lingering effects of the Green Revolution on small farming communities. So, the Green Revolution, as many of you are aware of, aimed at feeding the world through technology, hybridized crops, more synthetic inputs. But in the rice farming villages where my family lives, this approach led to the widespread adoption of hybridized rice cultivars that demanded heavy fertilizer use and pesticide use. So essentially pushing aside traditional cultivation methods, including the reduction of heirloom rice varieties. And seeing that gradual loss shifted my path towards deeper reflection. So much like the organisms in my compost or in the soil, I adapted in this new environment. So, but here the data that I'm working with tells a different kind of story, one of existence itself. So it speaks of life and death. It speaks of interconnectedness, symbiosis, predator prey. And throughout the session, I invite you to approach the content with curiosity. And when you're out there tending your land, hopefully you can use that same curiosity to guide you in understanding and connecting with your soil a little bit more intimately. All right. So, quick agenda: soil and soil health. I'm sure many of you already know the definition, but I just want to set the stage of soil and soil health, define soil health a little bit more. And I think there are some things that we need to keep in mind when we're talking about soil health: rhizosphere, phyllosphere, biological inoculants, and a case study with Stonecroft Farm. Thankfully, Glenn is here today. He's joining us. He's in the room, and so Glenn and Kathy are with Stonecroft Farm. So if you have any questions at all, please feel free to take advantage because they're here. Might as well take advantage of their presence. So the first three topics will provide a general overview, and it's the theoretical part of the session, and we'll set the stage for the more practical parts of the session, which are the bioinoculants and the case study with Stonecroft Farm. All right. Well, soil is an ecosystem. It is a community of living organisms merged with the non living components acting as a system. And the biology is at its core. So, to simplify, soil is made up of three main components: You have solid, you have liquid, and you have gas or air. So the solid components are made up of your mineral matter, your sand, silts, and clays, and your organic matter and biota, the living organisms. And then we have liquid and gas, the two components that I don't think we talk enough or often enough about. Productive soils contain a well structured network of pore spaces that regulate air and water movement. It supports root penetration and sustained microbial activity. And these interconnected pores act as ventilation channels within the soil. When water enters, it temporarily fills the spaces. It transports nutrients much like a highway system delivering food. So, water is basically a transporter of nutrients, an important component in soil. And as the water drains, air replacing it, supplying oxygen to microorganisms and plant roots. Soil air and soil water are interrelated and are dynamic parts of the soil. So Walter Yenny, a soil microbiologist and climate scientist, suggests to add nothing as a way to restore soil health. Here's another prompt for you folks. What does he mean by adding nothing? So feel free to type it in the chat. I'll check it out later. What does Walter Yenny mean when he's saying, in order to promote soil health, add nothing? So, soil is one of the most diverse habitats on the earth and contains one of the most diverse assemblages of living organisms. Here you have bacteria, fungi, algae, roots, protozoa. All of these organisms participate in one way or another in the evolution and formation of soil, especially the organic fraction. And it's important to recognize the dynamic events, the active relationships, and the players in soil. There are a lot of interactions that are taking place, different types of microorganisms, between the microorganisms and the plants, and between all the living organisms and their environment. So, soil is not a static thing. There is a lot going on that we have still have yet to uncover. John in the chat says synthetic fertilizers. When Walter Yenny talks about adding nothing, let me go back to that slide. Walter Yenny, what he actually means is building these pore spaces. That is what he means by adding nothing. Building these complex nothingness in the soil. It's another way to look at restoring soil health. You want complex pore spaces. And one of the ways to do that is to make sure that you promote decomposition, the decomposition process in soil. We're not talking about tilling to accelerate decomposition, but we're talking about allowing microbial activity or microbial communities to thrive in order to basically promote soil aggregation. And when you have complex soil aggregation, it'll naturally promote these pore spaces, which then allows water and air, the two components, not the two main components, but two components in soil that we don't talk enough about. So when water enters, air leaves, and when water leaves, air enters. These are very important components in soil, and promoting those pore spaces will promote these two components or the existence of those components. So now that we've established the definition of soil, let's describe soil health. What is it? You probably already know this. Soil health is the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. So this definition invokes the idea that soil is an ecosystem full of life. It needs to be carefully managed to regain and maintain its ability to function optimally. So how can we achieve soil health? You're familiar with the principles, knowing your context, keeping the soil covered, keep living roots in the soil, minimizing soil disturbance, planting diversity to encourage soil biodiversity, and integrating animals. To me personally, these set of principles, although it is moving you in the right direction, I don't believe it is complete. I feel like we can add one more very important principle, which is restore and sustain a thriving soil food web. With regenerative agriculture and gradual decrease of synthetic inputs, you really want an active soil community that will do those processes for you, meaning provide your plants with the nutrients that they require. And in order to do that, you need to promote decomposition. And so there are, um, I'm not going to go into too much detail, but let's explore what I mean by restore. Is it enough to diversify your cover crops and that will, in short time, restore the soil food web? It really depends. But in a lot of cases, um, it's going to take quite a bit of time. So this is the reason why I think we have to add the missing principle. Dr. Graham Sterling, who is based in Australia, inquired, why are most agricultural soils often bacterial dominated? They have lost their fungal biomass. Fungivorous arthropods, these are the organisms that feed on fungi. So if there's no food for them, they're not going to stick around. And the macrofauna population. I would even go as far as a significant reduction in the microfauna population, like your diverse nematode functional groups, your tardigrades. In general, these organisms are absent in our agricultural systems. So there are a whole host of factors combined that reduced and disrupted organism populations. We're talking about plant selection, our preferences for fast growing species, as an example. Simplified organic matter inputs. So, simplified organic matter, it's just going to promote more bacteria to thrive. It's really hard for fungal organisms to compete against bacteria because bacteria's reproductive cycle is much faster than fungal reproductive cycles. So if the organic matter is labile, more labile in the soil, meaning easily decomposed, bacteria is definitely going to be able to outcompete fungi. We have reduced soil habitat complexity. We're talking about the pore spaces that Walter Yenny was talking about as far as restoring soil health. We're talking about soil structure at this point. Physical disturbance, so overtilling, overgrazing, and increased traffic on soil. It breaks fungal networks. Fertilizers also inhibit fungi, so synthetic inputs can actually suppress decomposer fungi, which are the group of organisms that primarily rely on those recalcitrant types of organic matter, really hard to decompose organic matter as their energy source. We have tillage that disrupts mycorrhizal fungi, and we have phosphate fertilizers that also suppress mycorrhizal fungi. So high phosphate, it tends to reduce plant dependence on mycorrhizal fungi because why would the plant do the extra work to try and invite the mycorrhizal fungi to inhabit its root system when it's already getting the fast food from us? So the challenges involved in restoring a more balanced and fully functional biological community to agricultural soils should not be underestimated, as most soils used for agriculture have been under intensive management for at least 50 years. So this is what the studies have shown, which is 50 years, at least 50 years. That is a blip in the Earth's lifetime, and the extent of damage is staggering. So it's reasonable to ask whether these changes can be reversed. Now, Smith et al. Back in 2003 found that it took 9 years of traditional low input management to detect recovery. This is not complete recovery, that's just detecting some kind of recovery of the microbial community's fungal to bacterial biomass ratio at an intensively managed grassland site. Now, in this particular study, the restoration practices included hay cuts at different dates, specific grazing periods of cattle and sheep. They tested sections of of the fields that they were studying where they applied fertilizers and another section where they didn't apply fertilizers. They planted key plant species, manure applications, which had mixed effects as well. So it took 9 years for them to detect some kind of the fungal organism finally starting to pick up on its population growth. And then another study compared traditionally managed fields, TF, with agriculturally improved fields, IF, that ceased intensive management, found that restoring plant and microbial diversity in IF may take decades to a century. We don't have decades to a century. TF soils had higher organic matter, so those are the traditionally managed fields. It has higher organic matter, water retention, and fungal diversity, while IF soils were more bacterially dominated. And that study was done back in 2004. And to summarize both studies, conversion from an intensive fertilizer dependent system to a low input or organic system, which a lot of you folks are probably trying to transition to or trying to promote in your fields, occurs when nutrient cycling is primarily driven by soil biology, indicated by a significant increase in the fungal to bacterial biomass ratio. So, what that means is when you have I'll just continue with the next sentence here. A doubling of this ratio indicates successful conversion. A doubling of the fungal to bacterial ratio may not be something that you'll see in your lifetime. And this study says indicate successful conversion. I would even go, let's lower our standards. We can say, how about a balanced ratio as opposed to a doubling ratio? So, balanced ratio of F to B biomass, it should be enough to drive nutrient cycling and decomposition by soil biology. So, the study has basically shown that in order for those processes to actually happen, ecological processes such as nutrient cycling, where your plants are receiving nutrients from the soil without dependence on fertilizer inputs, that is indicative of fungal ratio increasing. If the current linear trend in unfertilized plots continues, full conversion may take 26 more years, which we don't have 26 more years. And more sensitive fungal species may take at least 30 years to reappear after intensive management has ceased. Again, we don't have that much time. So, why should we care? Why is it important to restore and sustain a thriving soil food web? Why do we want to make sure that it's not just bacteria thriving in our soils? Why do we want fungi? Why do we want all these other organisms that promote those predator prey interactions? Why do we want higher trophic organisms that can decompose the plant residue? When you're terminating your cover crops, you want microarthropods in your soil. You want fungal organisms that can essentially decompose the cellular structure of those plants, of the cover crops that you're incorporating. You want those organisms there. Why not just bacteria? Because the soil food web, when functioning optimally, supports the existence of all higher trophic life forms, including your plants, your animals, by providing these ecosystem services. You have retention and cycling nutrients to support plant growth. We're talking carbon, nitrogen, sulfur, phosphorus, um, water as well, energy flows within the soil, water regulation, storage, and purification, disease and pest management, toxin decomposition supports the production of food, feed, fiber, and fuel, and provision of health. Soil is a massive medicine cabinet. There are tons of different compounds that microorganisms produce, including antifungals, antibacterials, antihelminths, antimicrobials, anti-cancer compounds. And those compounds they produce due to environmental stresses, right? And when they produce those compounds, it's actually tied very closely with disease and pest management. They have the ability to manage pests and diseases. Plants have immune systems. They have the ability to essentially resist pests and diseases when they're given, when they're provided with that microbiome. All right, rhizosphere and the phyllosphere. So, what is the rhizosphere? The term rhizosphere was first coined by a German scientist named Lorenz Hiltner back in 1904. He was the pioneer in rhizosphere microbial ecology. It is the region of soil surrounding plant roots, or the region of soil that is directly influenced by plant roots. So we have some understanding of what happens in the rhizosphere, but we are still trying to figure out what is truly happening there. But it serves as an interface between the plant, the soil, and the microbes. And this interface is activated by the presence of microorganisms, bacteria, and fungi, along with their predators. So, this is our understanding of the rhizosphere today. How big is it? How big is this region of soil surrounding plant roots that the plants actually have influence on? It depends. The size can vary based on the plant species, the age of the plant, environmental conditions, the morphology of the roots. So a tree's root is different than oat roots, for example. So the zone of influence between these plants are going to be vastly different. It's been shown, though, that the rhizosphere can extend from 2 to 80 millimeters away from the root surface. So it's a complex system. So think of this as a 3D picture. So the interactions that take place inside the zone is influenced by the chemicals secreted by plant roots and the microbes inside these zones. So the soil here supports a dense population of diverse microbial community. The microbial community is more diverse and active as we get closer to the root zone than the area of the soil lacking plants or the bulk soil region. So a plant's microbial community can also vary as you explore different root locations. So say you have a potato plant here, let's just say, and then you have some kind of grass growing next to it, and a completely different plant, some kind of maybe flowering plant, whatever wherever your imagination takes you. Um, if the roots of this potato plant meet with the roots of this grass plant, the microbial community in that zone where the roots meet are going to be slightly different than the microbial community around in the area where these two plant species roots meet. So there are slight variations there. So it kind of makes you wonder what's happening. What is happening in the root system? What are these biological exchanges or microbial interactions occurring in the different parts of the root system? So this is why. So plant roots, they release carbon in the surrounding environment. And this process is called rhizodeposition. And the evidence of this has been known since 1894. Scientists have been studying this for more than a century. Yet here we are. We're still at the infancy of this research. It kind of makes you wonder who dominated the industry from then until now. When it was first discovered, B. Dyer noticed that organic acids were actually being released by roots of cereal crops, barley, oats, wheat. And one thing also to keep in mind is that there are many different types of root deposits. So there are many different carbon compounds that are being released by plant roots. You may already have heard of one of them called root exudates, but exudates is just one type of root deposit. Generally, though, root deposits are based on chemical structure, mode of release, so how it ends up in the soil, and its function. What do they do and what do they offer? So I'm going to talk a little bit about two types of root deposits. I'll introduce you to these two. And again, for each type of root deposit that is known now in soil science, you can go into a rabbit hole in understanding what these root deposits actually do. But the first one we're going to take a look at are called root border cells or RBCs. I'm going to just short form it because root border cells is too long. So RBCs are basically detached cells that come mainly from the root cap. So here we have a root, and then there's the root cap there, and then basically the cells that have sloughed off, they're called RBCs. Now, the root cap, it shows a rapid turnover of short-lived cells. The cells in the root cap are constantly being generated, specialized, and degenerated. So turnover rates of the cells in the root cap depend on the plant. RBCs are programmed to separate from the root cap and from each other as they reach the cap's periphery. Once they reach the edge of the cap, they're programmed to basically slough off, separate from one another. Once they shed into the soil, science has found that they are still viable. But at this point, they are referred to as border cells. They're viable because even after they've detached, the cells are still secreting enzymes and proteins. And they can survive for a few days in the soil, completely detached from the roots. Over 90% of cells that detach from the root cap survive. They don't die right away, at least for most plants. So, an example is field maize border cells. They were found to still be alive several days after detaching at some distance from the root cap. So the number of cells released from the root cap also vary depending on the plant. We have peas that can produce about 4,000 to 21,000 cells per day, cotton 8,000 to 10,000 cells per day, and field mustard, surprisingly, does not produce any border cells. Um, Plants that have a high propensity for mycorrhizal colonization, meaning plants that tend to build those relationships with mycorrhizal fungi, they produce more border cells than plants that don't, like field mustard. Regardless, that's a lot of organic matter that's being provided by the plant to the soil, which ties with how excessive tillage can destroy the channels that plant roots have created and the organic matter that they leave behind. Now, RBCs, they play an important role in plant and root growth. They help engineer the root's microbial ecology. So as they're detaching from the root cap, they're secreting proteins, they're secreting enzymes, they're feeding the biology in the soil. It's almost like the root is leaving food crumbs behind, attracting different groups of bacteria and fungi. RBCs also help roots experience less friction. When they detach, they also secrete mucilage or slime at the same time, lining that channel in the soil with a soft cushion for the roots to travel through effortlessly. They are also very important in plant defense. So not only do they secrete proteins and enzymes, they also secrete antimicrobial compounds. The second type of root deposit that I'm going to introduce you to, which many of you probably already know, are root exudates. Again, this was first discovered in 1894 by B. Dyer when B. Dyer noticed that organic acids were being exuded by grain roots. Um, Not just organic acid. So plants intentionally exude many different types of mixtures from the root system to deal with their environment. It's a sophisticated strategy employed by plants to survive in their changing environment, which means that plants can actually alter this process. They can decide, quote unquote, they can decide what they want to put out into the soil. So the plant, to some degree, it makes that decision. So if you think of your legume plant, that's one great example. How does the rhizobia know that the plant exists in the soil? There needs to be some kind of a biochemical exchange that happens between those organisms in order for that relationship to in order to initiate that relationship. Same with mycorrhizal fungi. How does the mycorrhizal fungi spore, if it is dormant in the soil, how does it know that there's a plant there that it can make that connection with? There's a chemical exchange that has to happen between the plant and the fungal organism. Because if you think about it, plants can't just get up and walk away, right? And say, I don't like it here. I'm going to go somewhere else. The soil is too wet. It's too dry. It's too restricted for my roots. I have way too much competition. They are forced to come up with all these different strategies to help them overcome to help them overcome different stresses. Now, exudates form the major part of root deposits, and within this group, there are various types of exudates. We know you probably have already heard of sugars as a type of root exudate, but it's more than just sugars. We have amino acids and phytosidrophores. Phytosidrophores are a class of chelator compounds that basically help sequester iron in the soil and make them more plant available. They help bind tightly to metal ions, like zinc as an example, and make them more available. And this is something that is common in grasses. Again, we have organic acids. We know this about the wheat, the barley, the oats. Vitamins. B vitamins are exuded by plant roots. Niacin, biotin, thiamine. Purines are also exuded. Purines, I couldn't find much research on purines, but what's out there is that it's used by the plant as part of its nitrogen assimilation. I believe this only happens with non-leguminous plants. And then there are enzymes that are released. Flavonoids. Flavonoids is a group of chemicals that legume plants clovers, your peas, your beans, they would exude flavonoids in order to establish that relationship with rhizobium, with nitrogen fixing bacteria. And flavonoids isn't the only type of chemical or class of chemicals that they exude. There's a cocktail of chemicals that legumes would exude in order to create that partnership with not with nitrogen fixing bacteria. There are proteins as well, and fatty acids. Fatty acids are exuded to promote and maintain mycorrhizal colonization. So plant roots, they play an active role in the rhizosphere. They can both be passive and active recipients of nutrients. They are not completely defenseless unless the soil has been decimated. But so in that case, they do need a bit of our help. They use various mechanisms to increase access to limited resources. And more often than not, they coordinate and associate with soil organisms to make that happen. So to some degree, they're not just sitting there waiting for things to happen. They make things happen with the help of soil microbes. So it turns out that plant microbial partners do not just exist around the root system. In the 50s, scientists began exploring microorganisms on leaves of plants, and it was F.D. Last, a British pathologist, who noticed a connection between pink yeasts and powdery mildew on cereal leaves, wheat and barley. He was able to isolate different fungal species from leaves of grains. So he drew an analogy with the rhizosphere and concluded that leaves have a phyllosphere. But the term phyllosphere has since been modified to include all aerial parts of the plant. So we have flowers, we have the stem, we have the fruits, we have the leaves, we have the seeds. Since the 50s, there has been a growing interest in understanding the complex microbial habitats and interactions found on all parts of the plant. So it's not just about the root system anymore. It's not about that zone of influence in the soil anymore. It's about the entire plant. Every part of that plant harbors a diverse array of microorganisms, and those organisms interact with the plant, affecting its health and functions. So, science around the microbiome has revealed that we have never been individuals. Each and every single one of us, whether you're a plant, a fungi, a bird, an insect, um, we all have a microbiome. We are shaped and influenced by the microorganisms on and in our bodies. And the term for this is called the holobiont. A holobiont is an assemblage of hosts and the many other species living in or around it, which together form a discrete ecological unit. So you're an ecology, I'm an ecology, your dogs, your chickens, your sheep, your cattle, they're all discrete ecological units, all interacting with one another. Lady Eve Balfour, one of the pioneers of organic food and farming, once said, "The health of soil, plant, animal, and human is one indivisible whole." All right, let's now move on to bioinoculants, the practical side of the session. So, how do you restore microbial life in the soil if the soil looks like this to begin with? If our agricultural systems have been intensively managed to a point that we're just really growing bacteria in our soils, how can we restore everything else? How do we get these organisms in the soil? Do you just start with cover plant seeds and hope they germinate? One of the best tools to use is high quality biodiverse compost. Now, the key word here, it's a tool. It's not the end all be all. It's a tool. You got to find a way to get the microbes in the soil, whether it's through your animals, your livestock, make them your microbial transporters if you can, or high quality biodiverse compost or something else. We gotta put these organisms back. Okay, so taking theory into practice, what does it mean by restoring the soil microbiota in the soil? As far as taking the context of composting, so we're just now into understanding what compost actually is and how it's a very useful tool in order to make this happen. Production of biologically rich and diverse compost. I will define that in a bit. What does it mean by biologically rich and diverse compost? Production of high quality liquid bioblends, like your compost extracts and teas, you need to have proper equipment to produce your teas and extracts to basically multiply your organisms in a liquid form. And you need the proper equipment to apply them into the soil. A lot of our farm equipment right now, they basically are built to deliver chemical applications. They're not built to deliver biological applications. And we're talking about not just bacteria, we're talking about the larger microorganism populations like your nematodes. And if you happen to have a good population of fungal organisms in your compost and they're producing that mycelium, the hyphae in the soil, you want to make sure that you can also proliferate them in your soil and monitoring the biology. Okay, so compost is a result of an aerobic decomposition process exhibiting amending, fertilizing character, and diverse soil organisms. So um, that is what compost is. So with composting, what we're doing is we're facilitating a decomposition process. We're providing a habitat to ensure that the biology growing and thriving in the compost are given sufficient amount of oxygen and moisture so they can effectively decompose the material. Let's break this definition down a little bit more. Exhibiting amending. So compost should contain compounds that when you put it out into the soil, those compounds will be stored. It will eventually be absorbed by microorganisms. It'll eventually be used up, maybe by the plant, but those organic compounds is it's your stored carbon, essentially, that microbes have built in the composting process. There's fertilizing character in the compost as well. So the compost should contain bioavailable nutrients so that when you put it out onto the soil, those nutrients can be absorbed by plants. It can be absorbed by soil organisms, basically used up right away, like your checking account, right? So if we're talking about carbon, if we're talking about these compounds, really, what are these compounds that are being taken up by the plant? So it's like going to the bank, taking money out of the ATM machine and going, I'm going to spend this money today, versus depositing into your savings account and using that later on when you decide to retire or for future generations, for your kids, for example. So that would be exhibiting amending, contains diverse soil organisms. So compost is not only about returning organic matter into the soil. It's not just about building up your organic matter, but it's also about bringing back the organisms to replenish the life system of the land. So think of compost as your pre and probiotic supplement for your plants or soil. You're essentially replenishing the microbiome of your land with high quality managed compost. So composting is an aerobic decomposition, meaning it requires air aided by the activity of a complex food web. So the key here, it's aerobic. As a composter, my job is to facilitate the biology that I'm growing or farming in my compost have adequate water and have adequate oxygen. Where do the organisms come from in a compost pile? They come from the organic matter that you collect to make your compost, as well as the environment where you're making your compost. So successful composting systems, they depend on creating conditions that are favorable to the growth and activity of microbial communities. It's like making a hearty soup, right? When I'm preparing my compost piles, I think about all the different ingredients that I'm using, much like you would if you're preparing your soil for your cash crops, for instance, right? What are the different diets that your land is receiving? And whether or not those food items are going to be for plant growth or not, right? And how is that diet going to affect the nutrition and the health of your cash crop? So in this case with composting, I also make sure how much of each I'm using, how much of each ingredient I'm using, and how they might interact with one another. Which organisms I think will thrive using those ingredients. So what should you get as a final product? If you're mindful of your composting process, you will be rewarded with a massive workforce that will help you in your farm. And that workforce is free. They're everywhere. You just have to learn how to multiply that workforce. Soil organisms are much better at growing plants than we are. Plants have established these relationships since time immemorial. And our job is to facilitate that natural relationship. It's a free team. You don't have to pay them, right? You don't have to provide them with the only benefits they get from you is a home that feeds and shelters them. And then let them do what they do best. Microorganisms are way better at gardening and at farming than we are. They know how to grow plants. Or maybe it's the other way around. Maybe it's the plants farming the microbes. But in any case, that relationship has been around way before we've existed on this planet. So the quality of your compost extract, sorry, I'm now moving on to liquid bioblends. I just gave you a general uh, very surface level intro on composting, but now we're moving on to liquid bioblends. Is the quality of your extracts and teas is determined by the quality of your base ingredient, which is your compost. If you don't have the biological diversity in your compost, you cannot expect your extracts and teas to have diversity. If you're using mostly manures, not enough woody material, you cannot expect diverse fungal biomass in your bioblends. You can't grow what doesn't exist. So why make extracts and teas? Why make liquid bioblends? Well, like compost, your bioblends will contain different substances that can be stored in the soil. They can fertilize your plants. They should contain diverse soil biology. Unlike compost, though, teas and extracts are distributed in a liquid form. So a liquid application can be advantageous because you get immediate nutrient availability. The soluble nutrients in bioblends are directly accessible to plants and soil organisms for immediate uptake. If there are nutrients there that are soluble and easily taken up by the plant, they can take those up right away. What compost, on the other hand, they provide a much slower release of nutrients. Now, I'm not saying that you shouldn't make compost if you are doing solid applications of compost. Continue with that process. This is just another tool in your toolbox, essentially, right? The microorganisms that depend on water films. So you get protozoas or protists, you get nematodes, potentially even microarthropods. These organisms, they depend on water films to move in and around the soil. And when you're distributing your amendments in the liquid form, it's just going to be a lot easier for them to find their way down. Flexibility in application, they can be delivered through irrigation systems or sprayed using equipment tailored to your context. It can make this approach scalable, adaptable, and suitable for your operation. It can be cost effective as well. So it's hard to procure large volumes of really high quality compost if you're not already making your own. It's a lot of money to buy large volumes of really good stuff, right? And on the other hand, you can just make a little bit, a small amount of really high quality compost, turn that into um, a liquid recipe, essentially, where you're putting your microbes in liquid form, and then you can combine it with cover cropping. So, which is a more practical alternative, potentially, depending on your context. Customizable recipes is another one. BioBlends also allow for easy manipulation of recipe. You can add different types of supplements into your BioBlends if you want. You can concentrate your biology in your BioBlend as well. In your extract, you can really make it as rich and as powerful as you want it to be. So what is the difference between an extract and a tea? A compost extract is simply just a process of extracting organisms, soluble nutrients, and organic compounds in water from compost. You're basically just taking your compost, you're putting it in a brew bag, such as this one, and then you're just extracting what's there into the water. And you can make this recipe as concentrated as you want it to be. Whereas a compost tea, it's the process of brewing an extract, you take that extract, you add microbial supplements, is what I call them. And I'm talking about things like fish hydrolysate, cold processed kelp, things that can grow your organisms, and you brew it for a longer period, and you're encouraging the growth of organisms in your tea. So let's compare the differences a little bit more. For a compost tea, it takes at least 24 hours to make. It's typically used for foliar application, although there are some exceptions where it can be applied to soil. Um, with teas, the addition of microbial foods in the beginning of the process will activate and multiply specific groups of organisms. We're talking bacteria, we're talking fungi if you have them in the compost, protozoas if they're there as well. Nematodes will not multiply in a compost tea. Since microbes like bacteria or fungi are multiplying, they're actively producing glues or biofilm, right? So when we're spraying the tea, bacteria or fungi will instantly adhere to the above ground surfaces of the plant. With teas, it's generally used to prevent disease causing organisms on the phyllosphere. And you can use it in soil. You can also you can use it in soil with organic matter levels of less than 3%. So foods that you've added into the tea, the dead organism bodies in the brew, ideally you're not causing more death in the brew. You're causing growth in the brew. They are essentially organic matter. And teas are very easy to screw up, especially with improper brewing conditions. If you have insufficient aeration, if you have too much nutrients in your tea, if you have shitty compost, they all lead to growing harmful organisms rather than the beneficial ones in the brew. And then you have the compost extract. Compared to teas, extracts are relatively easy or quick to make. It can take, I mean, it depends on your scale, it depends on your context, from 5 minutes to an hour, depending on the concentrated concentration preference that you want. So this is extremely customizable, the compost extract. The typical use for extracts is a soil application. So it's more of a soil drench, not a foliar application. The reason why a tea is a foliar application is you want those organisms that are producing the glues to adhere to the leaves, to adhere to the stem. Science have discovered that leaves, especially, they are um, uh, it's a habitat for all these different types of bacteria. Bacteria is the major group of organisms that tend to survive on. There are fungal organisms as well, but that's essentially what compost teas are made for, is to have those organisms stick because they're producing the glues in the tea. You want them to stick on the surface of the plants. So you're not adding foods to the extract at the beginning, but you can add it prior to your spraying, uh, prior to treatments. You're also right. So you're not growing the microbes in the solution because you're not agitating it for an extended period of time. Organism groups, they have different reproductive cycles, and you're not going to be. You may be growing bacteria, but you're probably not going to be growing fungi or protozoa in extracts. And again, with extracts, it's an effective means to get your predators into the rhizosphere. Predators, I'm talking about bacterial feeding nematodes, fungal feeding nematodes, predatory nematodes, protozoas. These organisms, they essentially feed on bacteria, they feed on fungi, and they promote nutrient cycling and decomposition process. You want predators in your soil, not just fungi, not just bacteria. You want those larger organisms and build up that soil food web community. So they should be in your compost if you're making compost. For extracts, you need to have sufficient organic matter levels in your soil. So you have to have organic matter level of 5% or more when applying extracts. It's completely pointless to apply the solution when there are no plants or organic residue present. You want them to be, basically, you want a seamless, thriving community. If you are using compost as your inoculant, that's your bioinoculant, you want those organisms to seamlessly flow from your compost through your equipment, your brew, your sprayer, down into the soil, and then them thriving in the soil. Finally, extracts are nearly foolproof. It's really hard to screw up an extract. So it's a reliable tool as long as you have the basic ingredient, which is a high quality compost. So, in summary, the application of teas and extracts is a means to effectively replenish the plant and soil microbiome for optimum growth and health. You have the tea for the phalosphere, and you have the extract for the rhizosphere. All right. Quick case study with Glenn and Kathy. They have integrated biological management into this. They are here joining us as well today. So hopefully Glenn and Kathy can be part of the conversation later on. So here they are. They are Glenn and Kathy Beaton of Stonecroft Farm. They both grew up on conventional farms. Glenn came from the prairies, and Kathy grew up here in BC. In 1977, they purchased a property and immediately started down the path of organics. Kathy came from a family of heritage turkey farmers, so she and Glenn decided to start growing turkeys in the early 80s. Unfortunately, last year, they had to significantly reduce their turkey population to a volume where they can no longer serve a lot of their customers because the next generation does not want to take over the business. And this change means that they can no longer meet customer demand during peak seasons like Thanksgiving and Christmas. They were always sold out. Every year, they sold out their turkeys during that time. So Glenn and Kathy, though, you can't take the farming out of their spirit. They continue to farm despite scaling back. They focus on chickens. They still have some turkeys. They also do blueberries, lamb, and they grow grain for their poultry. So their production size is roughly 70 acres. They cultivate their fields in addition to Glenn's best man's field. His name is Roy. They have been composting since 1994 and 95. The farm features a licensed abattoir, so it allows them to process their birds on site and ensure humane handling, which means they have to manage large volumes of high nitrogen resources. So, in addition to raising chickens, they have again blueberry slam. They grow grain to feed their birds, oats, wheat, barley, peas, rye. They also plant cover crops in the fall to help maintain a green living cover to protect the soil during our very wet winter season over here and to prepare it for their cash crops. So in late 2020, Glenn sought advice on improving his compost piles and learning how to produce liquid bioblends, specifically compost teas from his piles. He wants to know how can I take this resource and turn it into something even better. So the available resources on the farm are turkey parts, feathers, manure, and bedding from their animals. So these are all high nitrogen materials, and the only brown or woody component he had available at the time is the straw that came from his fields. They recognize the value of soil and they seek to optimize the farm's resources to cultivate grains for feed. All right. So the goal is to improve the farm's circular economy by optimizing how resources are managed. Now, the first entity are the primary products of the farm. Poultry, lamb, they're what makes this farm business profitable. They sell blueberries as well. They have cows, but they're not really sold for anything, as far as I know. They're employees of the farm. They help microbes. They help spread microbes all over the farm. So these entities serve as the source and destination for this program. Um, let's see. The raw resources themselves derived from the first entity, manures, feathers, turkey parts, and bedding materials, provide the base of the farm's fertility system. These are the available resources that will feed the micro workforce. Again, we are trying to uh, we're trying to multiply workers at the farm, microbial workers, because they're better at farming than we are. So, how can we take advantage of them? I know that sounds, can we create that relationship with them? How can we multiply them? How can we make them happy so that at the end of the day, they do the farming for us and we get crops at the end, right? But these resources, it's incomplete. The composition of the resources are mainly high in nitrogen, which will feed a lot of bacteria, but the fungal population will still be lacking. So we want to grow fungi, and this ratio here will make it very difficult for fungi to compete. So how can we improve this? The third entity in this program is resource and micro livestock management. Again, we want to grow diverse biology by ensuring that the resources are managed properly from assembly to maturation. And then the final part of the program, extremely important, is crop production and supporting soil health using their living compost, bringing in additional nutrients that will complement what's naturally available on the farm, performing analysis, equipment modifications, modifying soil management practices, anything that will support the crop that feeds their main products. Okay, so the foundation of the circular system is the micro workforce, right? The microorganisms breaking down the raw resources, turning them into living compost, and driving those essential processes like nutrient cycling and decomposition in the soil. They are the team that keeps things running and the ones responsible for helping your farm stay resilient, even when faced with potential threats and disruptions. They are a critical part of the farm business. And again, they're free. You can multiply them yourself, right? You just have to understand that it shouldn't just be bacteria and fungi that you're multiplying. Let's go beyond that. Let's go predators as well. Let's also multiply the organisms that feed on the bacteria and the fungi because they're going to continue that nutrient cycling process and promote decomposition and make nutrients available to your crops. So what did we do to improve Glenn's composting practices? We increased the woody or brown component. Luckily enough, Glenn had a few connections. He was able to connect with local arborists and get them to drop off their chips off on his property. He managed his turning times. How often did Glenn turn his compost pile? Is he aerating it? Given the high nitrogen component in his piles, it's crucial to ensure sufficient oxygen throughout the entire composting process. Nutrients can be lost in the compost if it is left at high temperatures for too long without aeration. Failure to aerate means that aerobic organisms will switch off, they will be deprived of oxygen, and these are the organisms that are essential to help retain nutrients and convert them into this rich material that are beneficial to plant growth. So, another thing too with using wood chips and how beneficial they are, some of you may not even have access to this, but any kind of woody brown material, if you can incorporate them in your compost, they can help maintain porosity in the compost, which helps aerate the compost overall. And I also encourage Glenn to move his piles outside. So historically, Glenn has been composting inside the barn. That's composting regulations here, which has an impermeable surface. That's just what the standard is you have to compost on an impermeable surface. But what we've done is after 5 turning cycles, I suggested let's move the compost outside because he's already gone through 5 turning cycles, decomposed the material enough that he's not going to get birds to come and, grab whatever animal parts from the compost. Um, so even after that many turns, his piles still heat up considerably because of the amount of animal parts in his compost. And it became time consuming, right? So this new strategy of moving the piles outside not only helps cool it, but he created a bait to lure organisms from the section of the farm, making his compost their new home. So we saw nematodes for the first time in his compost pile. He saw worms hanging out at the base of his piles. And so collecting data helped us understand where things can be improved. I love looking at data because that's my background. We want to make sure that the base ingredient, his compost, contains the organisms that he needs for the soil. So, as an example, this is a nematode comparison, by the way. The Christmas 2021 compost was analyzed at 2 different times. 3 months to allow for maturation was the first analysis, and then 8 months to allow for maturation. Because of the ingredients he worked with, it took 9 months after assembling the compost for bacterial feeding nematodes to inhabit that compost pile. It was way too hot for them. The amount of animal parts he has is just way too hot for nematodes to want to make that compost their home. So Glenn came up with a few different strategies. We talked about the data. We went, all right, let's make these changes. Let's see what we can do to improve the population. Compare those numbers with the Thanksgiving and the Christmas 2022 piles. We analyzed them 6 months after assembly and also roughly 4 months after assembly. And you can see bacterial feeders, the population was much better. So analyzing the biology helped us understand whether the changes he's incorporating into his management practice are working. And then here are the fungal numbers of his compost. Again, you can see how management of resources, the resources themselves they really matter. So let's take a quick look at how Stonecroft Farm integrated biological management into their farming practice. So when do they use these blends? When do they use these extracts and teas, and how do they incorporate it as part of their farming practice? So spring to summer, this is just the general summary of what they do. Cover crop termination typically happens in the early spring. Glenn trialed roller crimping as a practice for terminating cover crops, but he has since found that the practice can be quite ineffective, in his context. So now to terminate the cover crop, he uses minimal tillage. He used to do multiple passes of tilling, which led to high compaction, but he has since reduced the tillage passes in his soil. Liquid bioblend application. So at this point, he usually uses a compost tea shortly after cover crop termination because this helps accelerate decomposition. And the goal here is to stimulate microbial activity, ensuring that the plant residue becomes a nutrient source for soil biology, so that by the time the cash crop is seeded, beneficial microbes are already active, they already have that home, the cover crops that had been terminated, and they have now become Glenn's biological workforce to support crop establishment. And then he seeds with a liquid bioblend. So Glenn modified his sprayer system to inject a bioinoculant while seeding. Now, why do we want to do this? Microbes can impact germination of seeds in many ways. So plant growth promoting bacteria and mycorrhizal fungi, if they're present in the soil. Mycorrhizal fungi is not something that you can multiply in your brews. They are typically found in soil. Hopefully, they can speed up the germination process. They can also help seeds cope with cold stress or drought conditions, and they can help protect young plants from pathogenic organisms. Science has shown that disease-causing organisms are known to inhibit germination. So if your soil contains more disease organisms, then your seeds may not even germinate. Because why would the plant take that risk, right? So by inoculating seeds at the time of planting, the goal is to establish a strong microbial presence that will support plants throughout the growth cycle. And then biological applications during tillering and elongation stage. This is one of the most critical growth phases because what is happening at this stage in the plant? There is leaf development, so we need to support photosynthesis for growth. Root development is also happening at this time. Roots are anchoring the plant, they're absorbing nutrients, they're interacting with soil microbes. There's also stem elongation. The plant is extending itself, enhancing water and nutrient transport throughout its entire body. And there's also immune system development. So the plant is preparing itself for reproduction by developing disease resistance. Now, these are just some of the processes that are happening at this stage of the plant. The vegetative stage is extremely important in supporting the plant's microbiome. So beneficial microbes, they play a critical role in supporting all of these different processes, ensuring the crop is resilient and productive because what's in it for them, right? They want to survive too. They want to multiply. They want to grow, and they know how to make that happen, right? And synthetic fertilizer use. Glenn still applies synthetic fertilizers at the application rate of 8 kilograms per acre. alongside his biological inputs. So he's still using a little bit just to kickstart that biological system. Yeah, I want to see a decrease in this over time, but we'll see. So it still remains part of the fertility program. In the fall, he does have a good amount of compost available, so he will apply it in the soil. He seeds his cover crop blend as well, and he does liquid bio blend applications if the weather permits. So, what are the results so far? Behavior of water. So, in November of 2021, after less than a year of collaboration, I received an email from Glenn with the subject line "No ponding." So, he was very surprised. Many of his fields had always experienced standing water during the winter, especially with heavy rain events. And we live here on the coast, so it's a natural environment. And this standing water was significant enough that ducks would come and stay for the winter. Glenn's biological treatments began in March of 2021. By the end of that same year, the fields started allowing water to infiltrate and percolate. So this suggested to us that he is increasing the soil sponge effect. He's improving water infiltration. He's improving the ability for the roots to grow, nutrient cycling and retention. So despite experiencing a very wet winter, we had that atmospheric river happening here in BC. Despite the very wet winter, a lot of his fields did not have standing water for the first time in at least a decade. So at the farm, the fields were also dominated by fast-growing weedy species like cornspurry and smartweed. And these plants thrive in disturbed habitats. They signal the presence of poorly structured and poorly drained soils. The typical management before involved heavy equipment with multiple passes of the tine harrow in the spring once the weedy seedlings emerged, and driving heavy equipment in the wet spring it perpetuates compaction. But once they shifted to a biological approach, the weeds actually started to decline. And the grasses began to outcompete the weeds. So, how did this happen? Let's quickly talk about ecological succession in relation to the shift in the plant community. So, ecological succession is the natural process of change in plant and animal communities over time after a disturbance. If the disturbance removes almost all life and organic matter, such as after a volcanic eruption or an extreme wildfire that burns down the bare rock, it's primary succession. If some life and organic material remain, it's secondary succession. Over time, ecosystems can grow and become more complex, progressive succession, or decline in productivity, retrogressive succession. So, what happened at Stonecroft Farm? How did integrating biological management shift the plant community? They were here at the pioneer weed species community. The soils were continuously disturbed. It was compacted. Water can't flow, nor could air, right? So remember, soil is made up of solid, liquid, and gas. We need to make sure that the liquid and gas are able to flow seamlessly through into the soil because plant roots need oxygen and plant roots need water. There was a higher population of bacteria, lower populations of everything else. And nutrient availability is low in this part of succession. When they began to focus on soil biology, applying treatments, working to improve the soil structure, incorporating their cover crops, the soil food web began to shift. And this shift influenced nutrient cycling and availability, which in turn impacted crop growth and community structure. So they moved to over here. And this shift influenced, again, it's the interactions between predator and prey, the changes in the fungal to bacterial ratios, the restoration of decomposer organisms driving nutrient cycling in less than 2 years, ultimately leading to the grasses, early succession taking hold, successional grasses and perennial grasses started to take hold. And the new practice accelerated species replacement and vegetation stage. So going back to the slides an hour ago, where the question was, are we able to restore these biological processes? If our agricultural systems are bacterial dominated, can we restore these services that soil ecosystems provide us? Absolutely. You just have to work on improving the soil biology. Again, it is more than just organic matter, right? It is also your living organisms, as well as whether or not you are promoting or improving your soil structure in order for water and air to flow through. Um, since moving their compost pile outside, it also became inevitable for some plants to want to grow in their compost. So they had their compost placed on an area where it's next to a field that is growing clover. It's growing all these different plants, and somehow one part of their compost voluntarily grew clover. It was probably transferred there by the wind, and the root system had nodules, and when I squeezed them, they were pink in color. And this indicates to us that the compost doesn't just help spread fungi, it doesn't just help spread protozoa or nematodes, but also nitrogen fixing bacteria. They are multiplying nitrogen fixing bacteria in their compost. They are inadvertently spreading rhizobium in all of their fields, enhancing the nitrogen cycling process whenever they plant their legume cover crop. And of course, the most important entity in the nutrient cycling program are the crops that they grow to feed their animals. They have oats, they have barley, they have wheat, they have rye, they have peas. This is what we want to see. Though we had a really good weather last year, hopefully this trend continues. Tools used for monitoring field observations extremely important. If it weren't for Glenn and Kathy to continuously feed me with information, then I don't think we're able to be successful in so far anyway in moving or shifting their system to a much more complex system. Nematode analysis changes in management practices can directly influence the abundance and diversity of this group. They are consumers at various levels, they are predators in the soil, and they engage in nutrient cycling and energy transfer within the soil food web. So, analyzing nematodes was a way for us to understand whether or not we're building towards soil structure. Are we actually allowing water and air to flow through in the soil? They are microorganisms, but they are larger microorganisms. They need spaces in the soil and water film to move around and feed on their prey. So, understanding their population was again helpful for us to understand soil structure and whether or not we're moving in the right direction. Plant tissue analysis and soil chemistry testing. This was done through ANL. We look at OM%, we looked at pH, soil nutrient levels, and plant tissue levels. So we looked at what nutrients are exchangeable or available in the soil and what are the plants actually taking up. And at times, we found that there's discrepancy in what the soil chemistry is telling us and what the plant tissue is providing us. The levels may be present in the soil, but the plant isn't taking it up, or vice versa. So that's something that we had to really understand a little bit more. And not sure if we have the technology for this as well yet, to the point where we can truly understand what the plant is. It's a living organism, right? It's kind of like you asking me, I don't know, what did I eat today, right? What do I want to eat today? It's, or when do I need to drink water? Um, Protein analysis for grain or legume. The thing with this test is, although it's certainly valuable, so protein analysis is definitely a standard test. Um, but it doesn't take into account other nutrients, right? It doesn't take into account vitamins, trace minerals like zinc, iron, magnesium, complex carbohydrates, because they grow their grain for feed for their poultry. It's nice to have tests that tell us the other beneficial nutrients that animals also need, because I feel like protein analysis, yeah, again, they're valuable, but it kind of caters to the NPK, the nitrogen folks, the nitrogen fertilizer applications. It's very, it only looks at a very small fraction of what that seed contains, and that seed should contain other things as well.